Now we're going to talk about transcription. This is a DNA-dependent RNA polymerase responsible for this process. So transcription is the mechanism by which information in the DNA sequence is used to produce RNA. DNA is not converted to RNA by a chemical reaction. In DNA replication, the whole chromosome is copied in one continuous process. In contrast, transcription involves the discontinuous synthesis of many smaller products. Only a fraction of the total DNA in a chromosome, generally about 0.1%, is transcribed during the synthesis of an RNA molecule. Most mRNAs, which we'll discuss in a moment, are only about 1 to 10 kilobases in length, and the tRNAs are much, much smaller. So this is an overview of RNA synthesis. So synthesis of RNA is catabolized by a large enzyme complex called RNA polymerase. The polymerase copies the template strand of DNA to produce an RNA copy that is anti-parallel and complementary. The biochemical mechanism of RNA synthesis is very similar to that of DNA synthesis. The same basic mechanism is used in both prokaryotes and eukaryotes, although some of the post-transcriptional processing is quite different. There are 3 stages to RNA synthesis: initiation, elongation, and termination. The 2 strands of double helical DNA have different functions. Most DNA is double-stranded, but in a given region of DNA, only one of the 2 strands is copied during RNA synthesis. The coding, or the sense strand, has the same directionality and sequence as the RNA product, except it contains T residues, whereas the RNA product has U residues. The coding strand does not serve as a template for RNA synthesis. In contrast, the template, or antisense strand, serves as the template for RNA synthesis. The template is anti-parallel and complementary to the RNA product. And here we have an example of that. Here we have a figure that shows the coding strand of DNA. It is in black, has the same sequence as the RNA, with the exception of the T in place of where the U is in the RNA product. Not shown is that DNA contains a 2-deoxyribose and RNA does contain a ribose. The template strand in blue is anti-parallel and complementary to both the coding strand and the mRNA strand. So this slide just shows the flow, basically, of that genetic information from DNA, the template strand, the coding strand, the mRNA, which we'll find has words of its own called codons, and then the tRNA contain the anticodons that help bring the correct amino acids in when we're synthesizing a polypeptide. So the DNA template strand provides the information for the RNA synthesis. The flow of information from DNA to RNA involves the same language in that it is base pairing, or base per base. The flow of information from RNA to protein, however, involves a translation from one language to another. A sequence of 3 base units in the RNA is converted in a sequence of single amino acids in the protein product. So here we're going to discuss some of the critical characteristics of RNA synthesis. Once again, I can't stress this enough, only the template strand is transcribed to RNA. The coding strand, or the template strand, again, is the antisense strand, is not used for this purpose. However, the same strand does not always serve as the template. A DNA strand which functions as a template for one gene could be the coding strand for another gene or other genes. RNA synthesis can be initiated in specific locations without a primer. This contrasts to DNA synthesis, which requires a primer for initiation. Unlike DNA synthesis, there is no permanent separation of the two DNA strands during transcription. So after that DNA helix is opened up to be transcribed, it resumes or goes back to its helical conformation. Transcription occurs in an open bubble of single-stranded DNA, which is about 17 nucleotides in length. After transcription has occurred in a given region, the DNA strands rehybridize to form that double-stranded form. There is no editing or correcting of DNA or RNA, by nuclease activity. Transcription of RNA from DNA requires the following components. The double helical DNA to be transcribed, although it can use single-stranded DNA as a template. 4 NTPs, so we need those trinucleotides. And note, they are not deoxy, they are not, recall the deoxynucleotides are in DNA, and the ones with the 3' hydroxyl present are in RNA. We have a DNA dependent, and I'm sorry I'm forgetting and you can't see what I'm pointing at here, DNA dependent RNA polymerase. Okay. We have some other proteins which we're not going to talk about, but importantly we do have magnesium ions. So the mechanisms of RNA and DNA synthesis are quite similar. Again, RNA synthesis occurs on the template strand of the DNA, and it is transcribed in a 5' to 3' direction, just like DNA. So our template strand runs 3' to 5'. New RNA then is base paired as new nucleotides are brought in, and that RNA transcript then runs anti-parallel and is complementary to that template. Sorry, I've gotten a little scratchy here. This is just, a depiction of those new NTPs being delivered. Same mechanism as we've seen before, that 3' hydroxyl attacking the phosphate on that incoming nucleotide, and then the release of that inorganic phosphate. It shows the release of that PI, and we have an empty site ready for the next nucleotide triphosphate to come in. So the big question, how does transcription begin? We know that there are distinct sites at which the synthesis of RNA is initiated. But 2 questions. How does the polymerase recognize the appropriate start sites, and how does the polymerase introduce the first nucleotide triphosphate? And we're going to start with the first question, or the second question first, because that answer is actually quite simple. It always starts with an A. So that transcriptional start site accepts an A. So after the RNA polymerase recognizes the start site, then the appropriate nucleotide diffuses in, and its base hybridizes with the complementary base on the DNA template. We're going to see the consequence of no primer initiation with transcription. So the consequence of the initiation of transcription without the use of a primer is that the first nucleotide that is inserted retains its 5' the triphosphate component. Transcription will continue using the 3' hydroxyl, as we saw on the previous slide, to carry out the nucleophilic attack on the alpha phosphate of the next nucleotide. Okay. Actually, this one probably slide should have been before the other, but that's not the way they were numbered. So, at any rate, the first nucleotide in an mRNA transcript is an A residue. So this is a diagram depicting transcription in progress. So here we have that open, well, where's the open bubble? Open bubble. Here we have open bubble right here of DNA. So the 2 DNA strands have been unwound just in this region, and we have that growing nascent mRNA transcript, and you do see again that 5' triphosphate that will be retained in the final product. Okay. So here this is described as a DNA RNA hybrid mix that occurs through that transcription process. After the regions of DNA have been transcribed, the helix reanneals and reforms. So now we're looking at some promoter sites on DNA. The mechanism for recognizing transcriptional start sites is somewhat complicated. So we're going to take a look at 2 types of promoters, prokaryotic promoter versus a eukaryotic promoter site. Transcription is initiated at specific promoter sites on the DNA. One of the functions of the RNA polymerase is to recognize these promoter sites. So the DNA is carrying the message that tells that polymerase where and when to start transcribing the DNA and producing an mRNA transcript. So we'll look at the prokaryotic promoter first. There are 2 components which are generally centered at -35 and -10. In the -35 region, we have TTG, ACA. Okay. These are considered to be upstream of the transcriptional start site, which is at +1. The sequence at -10, TA, TAAT, is called the Pribno box. Okay. So many of these promoter regions have similar sequences of DNA, which are referred to as consensus sequences. So these are the consensus sequences at these 2 particular sites in the DNA. And these are the 2 most common sites in prokaryotic organisms. Promoters for genes differ markedly in their efficacy, meaning genes with strong promoters are transcribed about every 2 seconds in E. coli, and in contrast, genes with very weak promoters are transcribed maybe about once every 10 minutes. The -35 and -10 sites of strong promoters, have sequences that correspond most closely to the consensus sequence, whereas the weak promoters tend to have multiple substitutions at these sites. Also, the distance between these two consensus sites is very important, and their separation has an optimal number of nucleotides, which I think is around 17. Next, we'll look at a eukaryotic promoter, and it too has two consensus sites, one at -25 called the Tata box, and one at -75 called the cat box. So here's that cat. But this is the most common sequence in both of these promoter regions, or these sites. And again, here we have that +1, which is the start of, indicates the start of transcription, which of course is going to be an A in the RNA transcript. This is just a comparison of some prokaryotic promoter sequences at -10, and you can see that there is considerable variation compared to the consensus sequence of TAAT. So depending upon how close these are to the consensus, it will affect the strength of that particular promoter. So now we want to look at the question of how does RNA polymerase recognize the promoter region and the transcriptional start site. And we're going to look at the polymerase from E. coli. The RNA polymerase complex consists of 5 subunits. 4 of these are always part of the complex: alpha 2, beta, and beta prime. So those comprise the RNA polymerase. The holoenzyme, or the enzyme that is required to recognize the promoter, also includes the sigma subunit. Okay, so this is the holoenzyme, and this is the RNA polymerase. That sigma subunit is released after the enzyme starts making RNA at the transcriptional start site. So the sigma subunit then can be characterized as a transient component. It is responsible for recognizing promoter sequences. And the core complex can't start transcription at promoter sites without that sigma subunit. So the sigma subunit then contributes to initiation in 2 ways. It decreases the affinity for RNA polymerase for general regions of DNA by a factor of 10 raised to the 4th power, and it dramatically increases the ability of the polymerase to recognize the promoter sites. The mechanism by which the RNA polymerase recognizes promoter sites is very efficient. Rather than searching randomly, the polymerase complex slides along the DNA until it encounters a promoter region. The rate constant for binding of RNA polymerase to the promoter is 100 times faster than could be accomplished by repeatedly moving off and on the DNA. Once the start site has been identified, the sigma subunit diffuses away, and the core enzyme then completes the transcription process. So how does it know, that polymerase know when to stop? There is also a specific mechanism that determines the synthesis, when the synthesis of an mRNA will be terminated. In prokaryotic organisms, the signal for termination is contained in the mRNA product itself, and termination occurs when the RNA product forms a stable hairpin turn structure followed by a sequence of uridine residues. So we have the transcript, and then we have the formation, or the 5' end of the transcript, formation of this stable hairpin, and the U's at the end of that. So this is a signal for termination. So the formation of functional mRNA is a bit more complicated in eukaryotic organisms than it is in prokaryotes, because they are subjected to post-transcriptional processing. And there are three things that commonly happen to eukaryotic mRNA before it can function in protein synthesis. And those three are the capping of the 5' end of the RNA strand, removal of introns and the joining of exons, and finally the addition of a poly A tail. And we will look at examples of these. So here we have the coding region. So this is the region that is going to carry that information, or the codons, the words that will be read in order to produce a or synthesize, translate into a polypeptide. We have a 5' cap. The cap that's used in eukaryotic organisms is a 7' methyl guanylate. So all mRNAs in eukaryotes have this 5' cap. And the end of the message contains the poly A tail. There is a specific endonuclease which cleaves the mRNA, downstream of a sequence that reads AAU, AAA, and then a poly A polymerase adds about 250 adenylate residues. So obviously this isn't representative of the number of adenylate residues that are added to that poly A tail, or to form that poly A tail. And this is a diagram of the beta globin gene, and it shows the inclusion of the introns. So these are segments that are not needed for translation. And all eukaryotic genes have intervening sequences or introns of DNA within the gene that do not code for amino acids in the final protein product. So in genes that contain introns, the coding regions are referred to as exons, or expressed sequences. During transcription of genes that contain these introns, the entire gene is transcribed into mRNA. The primary transcript then is modified with the cap, the poly A tail, and the excision of the introns and splicing. So here we have the primary mRNA, it's been capped, it's got its poly A tail, and now we're going to excise those introns. So there is looping, there is splicing, so excision of those, and then the mature mRNA is produced, I guess. So let's take a closer look at an actual intron site and this consensus sequence for the splicing of the mRNA precursors. So here's exon 1, here we have an intron, here's exon 2, and so there are designated codes that identify the 5' splice site as well as the 3' splice site. And prior to this, there is a pyrimidine-rich tract. So those nucleotides that contain pyrimidine bases. And it is at those sites that indicate where the mRNA, mRNA, I can't talk this afternoon, gets spliced, and then those two exons are joined together. Our final slide is just a comparison of the transcriptional processes between prokaryotic organisms and eukaryotic organisms. Although most of the aspects of transcription are the same, there are a few significant differences. In prokaryotes, transcription and translation occur in the cytoplasm. Okay. There is one RNA polymerase that synthesizes all types of RNA, and there is minimal processing. So in this diagram, you can actually see that while transcription is occurring, we can also have translation starting right away. These little objects that look like mice with tails are the ribosomes, and the tail is the growing polypeptide. So these occur in the same compartment, and the processes can proceed at the same time, I guess is what I want to say. In eukaryotic organism, transcription and processing occur in the nucleus. Okay, so here's the nucleus. So transcription occurs in the nucleus. The processing to the mature mRNA that is capped, spliced, ready to be translated, then it is exported out of the nucleus into the cytoplasm, where translation occurs. There are many different RNA polymerases with different functions. We aren't going to go into all of those. And of course, the capping that is different as well. And again, translation occurring in the cytoplasm. Same process, but a different compartment. And that wraps up the lecture on transcription.